Hello, my dear students. I welcome you again in MVM Science and Home Science Teaching Learn. Today, we are going to discuss in this episode 6, paper number 503, Molecular Biology and Bioengineering. Today, we are going to discuss about unit number 4, Mutation and DNA Repair. And today's topic is we are going to discuss various types of mutations. Now, we all are aware about DNA replication, Mendelian genetics. Now, why I am taking this topic first? Because there are various reasons. Because if you are aware about these terminologies, it will be easier for you to understand other relevant topics in a proper manner. So, let's begin. Sorry for little inconvenience. So, to begin with, what is mutation and what are its various types we'll be discussing in this particular unit. So what is mutation? Mutation is the change in the sequence of nucleotide of DNA, right? Any undesirable change in physical, chemical or biological structure of any organism is termed to be mutation. Change in the sequence of nucleotide. We have already discussed in our previous lectures about the nucleoside and nucleotide. Nucleotide brings sudden change in morphological. Morphological means physical appearance of any particular organism. So any change in that characters. If such changes are heritable means it transmits from one generation to another, then it is called mutation. So, mutation is defined as any heritable change in the sequence of nucleotide of DNA. Now, organism with mutation is called mutant. Now, if any in particular organism mutation has taken place, then it will be termed as mutant, while the organism which do not have any kind of mutation is termed as wild type. Here in the figure, you will be able to see various types of mutation. These are all types of point mutation. Now, if it is normal and if there is certain change, then which type of mutation it will be termed, we will be studying. First is faulty proteins. If missense mutation is taking place, means in place of one protein, some another protein is being formed that is having some fault, that is known as faulty protein formation, which the mutation which occurs in this type is known as missense mutation. Then comes nonsense mutation. Whenever somebody is speaking something rubbish, we say that he or she is speaking this nonsense. But in such mutation, what is occurring? Incomplete formation of protein is there during the process of replication. I know we have not discussed in detail about process of replication, but during the process of replication, if stop codon is generated, stop codon is UAG comes in the middle, then the process of replication will stop, and that will lead to the nonsense mutation. Next comes silent mutations, means it is not being expressed, but it has already happened. That type of mutation is known as silent mutation, and another one is wild type, that is normal. Normal was this case like THC and ATG, right? Then it will be forming tyrosine, that is normal protein. But here you can see that instead of T and A and T, protein is formed, but something is changed. So normal it is there, but mutation has occurred, so silent mutation. What is the case over here? Instead of G and C changes there, so UAG. So stop codon will be there. And here faulty protein AAC is forming, that is missense mutation. So these are the types of point mutation. Now let us understand what is point mutation. Point means only one single place change will be there, that will be leading to formation of some different kind of protein than the normal protein. This is termed as point mutation. Only one point. There is change or mutation in only one single point. Now let us discuss all this in detail. First is a silent mutation. It is known as the neutral mutation. We cannot see the mutation actually, but there is mutation inside it. It is the mutation in which mutated codon codes same amino acids as the original codon. So amino acid formation will be same, but some position change will be there. Since the amino acid is same as original one, it does not affect the structure and composition of the protein. Silent mutation causes phenotype of bacteria remains similar to that of wild type. Phenotype and genotype we have already discussed in our lecture. So phenotype, physical appearance will not be changed. Next is missense mutation. Missense means it is not making any form of sense. In this mutation, mutated codon codes different amino acids. Since new amino acid coded by mutated codon is altered, the protein form is also altered. Such protein can be less active or completely inactive. Such proteins will be formed which will be either non-active or inactive. If altered amino acid lie on active site of protein, then such protein becomes completely non-functional. So that protein has formed, but it is of no use. Next is nonsense mutation. Mutation in which altered codon is stop codon or chain terminating codon. Such mutation is called as nonsense mutation. Nonsense mutation causes incomplete synthesis, means nothing has completely been formed. Some incomplete proteins are formed that are called as nonsense mutation, or they are generally non-functional. This brings greatest change in the phenotype of an organism. If such type of mutation takes place, then it will form a greater change in the phenotype, that is morphological characteristics of an organism. Now, next topic we are going to discuss is frame shift mutations. Now, let us understand the word frame shift. Frame means suppose you are viewing something, whatever you are viewing or whatever in optics you are seeing, that is your frame, right? Whatever you can view. Suppose you can see a wall next to you, so that is your frame. So whatever things will be present inside that wall, that is termed as your frame. Now from that wall something is shifting from one place to another, that is termed as frame shift mutation. If it occurs as a result of either addition, if you are taking an example of wall, you are adding one flower vase, right? So you are adding something to that frame, it is addition. If you are removing something from, for your example, you are removing the wall clock, then you are deleting something from the wall. So such type of additions, deletions, right, these are termed as frame shift mutations. It results of addition or deletion of nucleotide in the sequence of DNA. Addition or deletion of nucleotide causes shift of the reading frame. Whatever we are seeing inside that, there is some shifting is there. In an mRNA, each codon is represented by 3 bases. Without punctuation and insertion or deletion of nucleotide changes the entire frame. So frame shift mutation brings greater phenotypic change than point mutation. So here you will see that in terms of point mutation, you will be having some more effects in frame shift mutations. Insertion or deletion of 1 or 2 base pair of nucleotide causes shift in the frame. However, insertion or deletion of 3 base pair adds or removes a whole codon. Suppose it is AAC and only C is being added, so it will be now AACC. But if it is AAC and again CAG is being added, so something fruitful, something meaningful is being added, or something is being removed or deleted. This results in addition of removal of single amino acid from the polypeptide. Mutation in which altered codon is stop codon or chain terminating codon. Such mutation is known as nonsense mutation, causes incomplete synthesis. Now, insertion or deletion of 3 base pair adds or removes a whole codon. This results in addition and removal of single amino acid from a polypeptide chain. So how it is different from nonsense mutation? In nonsense mutation, something is either removed or either added, which is making no sense. But in frameshift mutation, if it is being having some sense, see, insertion or deletion of 1 or 2 base pair of nucleotide causes shift in the frame. So now reading changes. Suppose it is AAC and you are adding CAG. So now it is reading AAC, CAG. But if it is only adding AAC and we are adding CA, then now it becomes either AAC or it can also read CAC. See, here is the figure. You will be able to easily understand what is addition and deletion. Original codon of an amino acid. All are coding histidine. But see, if there is some insertion of single nucleotide, so now the change is that, suppose it is CAT, 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 CAT. So up till now it is histidine. Now only one addition is there of A. Now it is coding for 309 serine serine. One is being changed. So insertion of single will form an incorrect amino acid sequence and functioning of proteins will be hampered or it will be incorrect. So this was basic idea about two main types of mutation. One was so point mutation and another was the frameshift mutation. Now today you might be thinking that very small topic is there, but there was some reason for taking this small topic in between, so you all can understand the process of transcription, translation, protein coding in an easier manner. Also process of DNA replication can be understood in an easier manner. That's why we have taken this small particular topic in between. Two different types of mutation. Now let us understand something else. Mutations, if they are occurring, in 90% of the cases they are causing some or the another harm to an organism, but in few of the cases mutations are very much helpful. As for example, if somebody is suffering from some genetically disordered diseases, right, for generations, 2 3 generations, but somehow some mutations are taking place and that person or that organism is coming back to normal because of the mutation. So this is the positive effect of mutations. Now in studying genetics, where we are going to use this mutations, for example, we want some organism to produce some particular product. So why that organism will produce that particular product? Because we want it. That organism does not require for its metabolism. So at that time we need to create mutations, we need to genetically modify it so as that particular organisms forms our desired products. So here we can use mutations. It is very much uh, uh, required for us to understand that why mutations are taking place and what are the causes and results of the mutation, right? So I hope you all search about mutations, various types of mutations, chromosomal aberrations, and why they are occurring and what can be used to control them or stop them. So this was just an overview of different type of mutation. Hope you all enjoyed this video. Hope you all understood it. For more queries, you can always contact your teacher. So with this, today we are ending our topic, but this is not the end. This is the beginning of the study. So start studying. Stay home, stay safe, and best of luck all. Thank you very much.